Embryo of Dicot seeds. A seed is a small embryonic plant structure which is capable of growing into a new plant. A typical seed has three basic parts: an embryo, storage of nutrients for the embryo, and a seed coat. All seeds are different in size, shape, and color. Under proper condition for germination, the embryo develops into a mature plant. It is nourished by the food reserves in the storage structures called cotyledons. The embryo has one cotyledon in monocotyledons and two cotyledons in almost all dicotyledons. Materials required: watch glass containing seeds of bean, gram, and pea, forceps, needle, 3 small beakers containing distilled water, and a dissecting microscope. Procedure: put the seeds pea, gram, and bean into 3 different beakers containing distilled water. Soak the seeds overnight. The seeds imbibe water through the micropyle when soaked in water. Take 1 seed from each beaker and put it into the watch glass using forceps. We can see that the bean has a kidney shape. A gram and pea are round in shape. On each seed, we can easily identify a scar called hilum. The seed is attached to the inner margin of the fruit by means of the hilum. Taking a look at each seed carefully, we can observe a micropyle on the seeds of bean, gram, and pea. Water is absorbed via the micropyle during the germination of the seed. Carefully remove the seed coats of each seed using forceps and a needle. Place the seed onto the stage of the dissection microscope using forceps. Separate the 2 cotyledons of each seed using forceps and a needle to locate the embryo between them. Examine each part of the embryo of the seed through the lens of the dissection microscope. An embryo consists of an embryonal axis to which 2 thick fleshy cotyledons are attached. The cotyledons are food storage organs. One end of the embryonic axis lies enclosed between the 2 cotyledons and is called the plumule. It develops into the shoot. The other end of the embryonic axis protrudes outside the cotyledons and is called the radicle. This develops into the root. Since 2 cotyledons are seen in bean, gram, and pea seeds, they are dicot seeds. Precautions: seeds should be soaked overnight in distilled water. Be careful that the seed is not damaged when removing the seed coat.